We're going to be spending a little bit of time here this morning, and I'm going to be talking to you about the why, the what, and the how of genomics technology. But before I get started with the presentation, and let me know if it's too loud, okay? I sometimes don't need a mic. I speak quite loud, but let me know if it's too loud and I can kind of lower the volume a little bit. But before I get started on the presentation, I'm going to give you a little background about myself and what I do today. I know Colin touched on a little bit. I'm an animal geneticist by training, and so when I did my PhD, I was fortunate enough to be part of the group that developed the very first bovine chip, the bovine 50K, and then that catapulted the work for us to publish in science. So that was a very big part of the community, the scientific community, which was decoding the bovine genome. So that was very good for the scientific community. And that set the base for everything that we're seeing today in terms of the implementation and the use of genomics in animal breeding. Shortly after I left the academic world, I had a chance to work for a seed stock company, and I got to see firsthand what it was like trying to convince breeders to use genomics technology, and at the same time we were trying to work out the development of specific selection indices, or indexes as I like to say, for breeding programs. And this was back in Canada at the University of Alberta. And before I get too ahead of myself here, I was actually born and raised in Brazil. Dr. Cliff Lamb yesterday said he sounded funny. He was from Zimbabwe. I don't know if I sound funny or not. I moved to Canada, so you probably hear a Canadian accent more than anything else. So I'll sometimes throw out the A's and abouts. But ever since moving to Nebraska about 6 years ago, I think I've lost the accent, and I think it's diluted over time. But if I'm talking to my Canadian friends, you'll definitely hear it. I was working for a seed stock company back in Alberta, Canada, and it was one of the very first seed stock companies that invested in genomics technology. They started out with simple parentage testing, and I'm going to talk about parentage testing throughout the presentation as one of the strategies for anybody that wants to get in the game. It's a really simple but very powerful test. And looking back, that was the role that exposed me to the important topic of communication and training of those entrusted with using the technology. So, and up to that point, I didn't really have a lot of experience with it. Daryl and I were just talking about this shortly before I came on stage. I really believe that we need more ag communication classes for master's and PhD students, particularly those with a technology background, to be able to communicate that use properly to everybody using breeders, seed stock and commercial producers. And the way that I decided to do that, or to help out in doing that, was to start my own company. It's called AgFRONT, and it's really a digital company more than anything else. So after working for a biotech company in Nebraska, I decided that if I really wanted to move that dial a little further, I needed to do it a little differently, and the only way for me to do that was to become, what everybody tries to say is an entrepreneur. And farmers and ranchers are the original entrepreneurs, so I pick their brains. I have picked their brains for years now on how to do it. So I decided that that's what I needed to do. I don't know how long I'll be doing this for as long as I can, for as long as God willing allows me to do that. So yeah, it's a digital company. I built my website, learned how to build it, and on that website I publish content related to actionable tips on the use of genetics, genomics, and biotechnology. So I allow people to subscribe to the list, and in turn I send them information whenever I can. So that's how I'm going to do it, right? Everybody wants to feed the world, and everybody has different hows, and the how for me is through training. And I'm going to harness the power of technology, no pun intended, to do that. And because the world is digital, everybody today make purchasing decisions or training decisions, or they learn things, they go online, right? They go to Google, they go to Yahoo, and then they type in certain words, and then they find websites and information related to the topics that they want to learn. So after traveling around for years, I had a former colleague friend of mine that's there, Jill Ginn, works for GeneSeek. So say hi to Jill if you can. And so, you know, we traveled a lot. In my role, I travel a lot, and the company found out that I had a Brazilian passport, and so they said, okay, after you're working in the U.S., let's move over to Brazil and Latin America and trying to see how far we can go, because the thought behind that, and I want to make sure that everybody's clear on it, everybody's very competitive when it comes to their products and services, but we have to be aware that we can't sell something if the customer doesn't know how to use it, right? So particularly a breeder that's using genomic enhanced EPDs or is investing in genomics to have genomic enhanced EPDs, how are you going to sell a bull that's been genomically profiled if your customer doesn't know the benefits of genomics? And so this day and age, communication and education is your biggest marketing tool. So that's the biggest way that you're ever going to get that client, is if you educate and help train that customer and help add value along every step of the way of your sales process. So for the longest time, I think society taught us to kind of keep that information close to our chest, and to a certain extent, sometimes that's what you need to do, particularly if it's something that hasn't been patented or in that level. But if you want to gain that customer, if you want to gain that trust, you have to learn how to communicate that information. You have to teach the customer how to use it. So that's my philosophy, and that's the reason why I started AgFRONT. So here's the website. The presentation is going to start out a little slow because I want to cover the basics and the base of genetics and genomics, and I'm going to talk about what each of those terms mean. So this is the website. I am so proud of this website. I built it myself, I learned how to do it, and you cannot be in the middle of technology this day and age without having a website. And I say this for myself and for everybody out there in the livestock industry and the ag industry in general. You have to have a website. Okay, that's very, very important. Tom and I chatted a little bit yesterday about social media and the role that social media has in communication of whether it's technology or your products and services. It's massive use of social media today. Facebook, for example, has 2 billion active users. 2 billion active users. So you need to start thinking about how you're going to tap into those users for your sales, right? So I know a lot of breeders already use Facebook. They've got a Facebook fan page. And they publish, content or posts every day or every so often. So you have to learn how to harness the power of social media for your advantage. Sometimes we spend too much time on social media, and we become, not social anymore. So it's the oxymoron. It's really, I don't know how we got to that level, but we can't go back anymore. So we have to learn how to harness that properly. So if you have a chance, please visit the website. So it's www.agfriend.com, and there I publish content, no nonsense. And people that know me know that I don't like to waste time. Ain't nobody got time for that. I'm very to the point, very direct. So take a look at some of the articles and the content. One in particular, and that's the one that the invite for this conference came about. It's called The Future of the Breed Associations. And this article really ruffled some feathers, maybe not feathers, but tail hair, because I talked about the role of the breed associations in breeding improvement programs compared to what it was years and years ago. Now, breed associations are no longer just registration companies, right? They're now in the middle of technology. They have to be in the middle of technology. They're part of the breeding improvement programs. Now, and with that comes specific responsibilities with communication and training, and also investing and encouraging their members to continuously collect data, right? In the age of genomics, phenotypes are probably most important. They will not go away. The need to collect data will not go away. So that's one of the points that I want to make throughout the presentation is data collection. So take a look. I think it got some shares. People liked it. Some people didn't, because some of the genomics companies probably didn't like reading this, because what's happening now is a shift. Right? Before, the breeders were a little at the mercy of genomics companies when it came time to incorporating the DNA information into traditional genetic evaluation, and they held all the pieces to the puzzle, and now not so much. Now the breed associations have access to the methodology themselves. So today, for example, BBU has access to the GBLUP methodology that John Janot has implemented. John has been at this convention a few times, I believe, talking about this. So the power of the breed associations, that is definitely increasing. Okay, it's a big player. So breed associations have to be aware of this, and they need to kind of play strategic. Sorry, my computer's going off here. Strategic, I guess, moves when it comes to continually to be at the forefront of their industry. So just be aware of that. As your breeders, you're part of the breed association, so make sure you're part of that as well. There are going to be several definitions of genomics throughout the presentation. They're all correct in their own way. If you were to ask a geneticist what genomics is you get an answer. If you were to ask a genomicist, you get another answer. If you ask a breeder what genomics is, you get another answer. The answer that you get is highly correlated with how they see themselves as the user of genomics. And I'll start by defining some of the differences between genetics and genomics, particularly because many still use these terms interchangeably, and they're not exactly the same thing. All right, so the first thing that I'll say is that genomics and genetics, they go together like peanut butter and jelly if you're in the United States, or peanut butter and jam if you're in Canada. Right? So the two terms go together. You cannot talk about genetics these days and not talk about genomics, just like you cannot talk about genomics and not talk about genetics. So the two terms go together. Now, genetics tries to understand how genes, which are the basic units of heredity, influence specific traits. Right, and how information is passed from generation to generation through DNA, the molecule that makes up the genes. Now, genomics has gone mainstream, and the term has been used more and more now, but it's really a field that's interested in studying the entire genome, its entire sequence. So advancements in computational biology and DNA sequencing really allow the field of genomics to make significant contributions to the rate of genetic progress in many livestock species. So with genetics, you are used to hearing that term with the term EPDs, right? Here in North America, use EPDs. In Latin America, it's EPDs. In Australia, it's EBVs, right? But then we're going to see the difference between the two. So EPDs, that's the genetic merit of the animal as a parent, okay? Some other basic concepts. So genes are the basic units of heredity, which consists of sequences of DNA in a specific location on the chromosome. Here's a picture. I've got a picture of the cell on your right. Nucleus, that's where we harbor all the chromosomes. If you were to kind of unravel the shape of a chromosome, this is what it would look like, a series of A, T, C's, and G's, which are the nucleotides. And a gene is basically just a sequence, a DNA sequence at a particular place in the genome. So sometimes we talk about genes, right? Those are the genes on a particular chip, right? Or we talk about genetic markers, right? SNP, S and Ps. So we use those sort of interchangeably for the sake of simplicity as we try to explain what the technology does. This was a screenshot that I got from the website, BBU website, and just released, essentially saying that Beefmaster has released genomic enhanced EPDs, so GE EPDs. This was about a year ago, so June 6th, so just a little over a year ago. BBU released those. So that's a milestone for the breed association. And of course, I don't have to say this again, so make sure you utilize and leverage that opportunity that you have. If you're a breeder and you have not looked into using Genomics Free Breeding Improvement Program, I would urge you to start looking into it now. Many other breed associations have. The very first one was Angus, and this was about 7 years ago. 7, 8 years ago. It was a different model back then. They were the very first one. There's some bumps in the road for sure in the implementation process. Part of that was communication. So every time you're the first in anything, you take some of those bruises, and hopefully people will learn the lessons from you, and so they do a little bit better after that. So breed associations that started incorporating genomics after Angus learned some lessons, and so they didn't make the same mistakes. Okay, so that's good. You're not going to be as bruised up as some of the first ones were. What I do want to be very clear about what genomics does not, okay? And I alluded to that early on in the presentation. Now, genomics does not replace the need for data collection. That will never go away, okay? It's become more important now than before. And it does not replace the need for EPDs, expected progeny differences. Early on, and I like this graph a lot because it shows the technology adoption life cycle. And you see expectation over time at the top, and you've got the adoption rate at every stage of along the timeline there. And you look at the peak of inflated expectation, right? High there. Every new technology that's out in the market, we think that's just going to solve all of our problems, right? And right around that area, at that time, I don't want to blame anybody, but the community as a whole, and this included, some scientists and some genomics companies, we're thinking maybe genomics is this, and we don't really have to start collecting any more data, because that's a painful process. I think if you were to ask, I don't want to speak for yourself, but for you, but if you'd say one of the things that you don't want to do anymore is probably collect data, would you say that is it a painful process? Yeah. So everybody is trying to say that you don't have to collect data anymore, just use genomics, buy my product, and voila, you'll have, it's magic. And it wasn't like that. And another big problem at inflated expectation was the fact that the message out there was that you didn't have to genotype and invest in specific breeds. You can just pool everything, and a little bit here, a little bit there, and that's enough to have a really good sound prediction equation, right? So you can predict those markers using a little bit of the some of the animals that are already existing in a database, and that's not true. Okay, so we figured out that predictions were breed specific, that if we wanted to predict in Red Angus, we couldn't use Angus. If we wanted to predict in Angus, we couldn't use Red Angus. If we wanted to predict in Beefmaster, we couldn't use, Brahman or Hereford. We needed to be specific. So, so once we squared that away, then we hit the trough of disillusionment there, and then we're now on the slope of enlightenment. So everybody's a little bit happier now when it comes to implementing genomics and understanding what it actually does and some of the strategies behind genomics. But it took a while for us to get there. Obviously, we never present anything that doesn't work, right? Most of the time we just, our presentations are filled with things that work, and nobody talks about the things that didn't work. But there's a lot of blood, sweat, and tears over the years in trying to fix those things, and a lot of people came together to fix those issues. Genomics is a game changer technology for any organization involved in the food production industry. Okay, and we're going to see some of the specifics of each of the players now being part of the livestock industry and how their power has increased over time. I like to say, and I have a slide here that's going to showcase that, and some of the analogies that I make in some of my presentations have to do with building things and speed, right? Fast cars, and it jives well with the mostly male audience. They seem to like speed, fast cars, and they seem to like building things. So I talk about the foundation, pouring the foundation first before you build the first floor, and talk about Nitro Boost, which is really what genomics is. As it's another way of looking at genomics. The Nitro Boost for Breeding Improvement Program. This is a slide here that gives an overall picture. If you were to remember nothing about the presentation, keep in mind that this is probably going to give you an overview of each of the players in our industry and how they interact. So you've got a producer. Let's see if I can use this. Producer that's submitting data to the breed association. So you submit contemporary group information, individual records, pedigree records, and progeny records. The breed associations work very closely with the genetic evaluation companies. Some breed associations actually, as part of their subsidiary, as if you're Canadian, will have their own genetic evaluation company in-house. So you have Angus has AGI, and ASA, which is American Simmental Association, has IGS, International Genetic Solutions. And BBU, you work along with John Janot from Livestock Genetics. So they have to work very closely together, the breed associations and the genetic evaluation companies. And right on your right there, you've got laboratories and animal health companies. Now, you have to work very closely with your laboratories of choice, particularly now in this day and age with genomics. Sample submission, information submission, all of that needs to be well thought out, all of those strategies of information and sample flow, because if you miss a step there, that's a big problem. And that's one of the things that early on we struggled a bit, right? We needed to, it took a while to get that sorted out with the breed associations and with the breeders. It's always a process. It's always going to be something that needs to be tweaked. And as you invest, as a breed association or the associations invest in software, sort of a portal that allows for submission of sample information to be done via the internet, via your website, the easier it's going to be. But just like anything else, it takes time for people to get used to that and to submit information online, whereas before they were doing that through paper. So yeah, work very closely with the laboratories. The laboratories also work very closely with the technology companies. So you may or may not have heard of Illumina or Affymetrix or Thermo Fisher. So those are the companies that develop the equipment that the laboratories use to process the samples. So again, work very closely with them. And all of that information, so once a producer submits data and sample to the breed association, maybe directly to the laboratories will process the samples. We'll send the raw data to the genetic evaluation companies. So if you remember earlier slides where I talk about the bases, A, T, C's, and G's, that is the raw data that we get out of the equipment that the technology companies create. Right, so that's the raw data that is sent to John Janot, and then John uses a methodology, GBLUP, to incorporate that information into the genetic evaluation. Okay, so all of that is done behind the scenes. You don't see that happening, and it's not trivial. It's very cumbersome. So it took a while to get to that level of pressing the enter button, if I could say that. And once that information is submitted back to BBU, then you receive an individual animal report with, GEPDs. Back in the day, I talked about some of the inflated expectation peak there. The genomics companies were submitting what we call molecular breeding values, right? They were doing all of the analysis themselves, right? The genetic evaluation companies weren't there yet. And at the time when the animal genetics companies were doing that, they were submitting reports that were molecular breeding values. They were not EPDs with genomics information. It was basically just a standalone report, 1 through 10 or percentile. And that's not the way to talk to the breeder if you want genetic improvement to really happen. You have to incorporate that into the traditional genetic evaluation. And that's why today you have GEPDs. You don't have an EPD and then another genomic number. Everything is in one. And that's the way to do it. But before there were two separate things. It caused a lot of confusion. Breeders didn't know how to use, the two separate numbers. And so we squared that away, and now it works as it's just GEPD, just one number. And you probably notice, if you were to compare, before and after genomics, the increase in accuracy, right? Because that's what genomics does. One of the things that genomics technology does is that for lack of better wording, it looks under the hood. It really sees what the animal's genetic merit really is. Because up until this point, with just a traditional genetic evaluation, if you're a young animal, you're just getting your parent average. There's no performance information associated with you. You're young, so you're using your parent's average. And we know that even full SIBs will have different, all of them will have different DNA markers, and it's not their parent average. They're not going to have the same EPDs at the end of the day. They're all different. Right, so genomics really looks under the hood for each of those individual animals and allows the stratification of individuals that before were on the same level of genetics for that stratification to happen. And at the very end here, I've got producers interacting with AI companies. Many AI companies now are in the middle, they have to use genomics, right? So they've been kind of slow to be in the middle of it. And I don't know if you know of a company called ST Genetics, formerly known as Sexing Technologies. So they were not even an AI company back in the day. They licensed a semen sorting technology from the USDA years ago. And because some AI companies were slow to get in the game of using genomics, this company called ST Genetics took advantage of that and said, okay, we're going to start purchasing bulls. We're not even an AI company. We're going to start purchasing bulls that have been genomically enhanced. And they now have, and don't quote me on this, it might be different, 30 out of the 100 top bulls. So they now they vertically integrated. Patents don't last forever, right? Semen sorting technology. And they now became an AI company, competed with some of the really old AI companies out there, ABS, Alta, et cetera. So now ST Genetics is an AI company. It's a pretty big one. Let's talk a little bit about simple versus complex traits. I've talked a little bit about EPDs, but I want to go back to the basics. So the reason why we use EPDs is because we are dealing with complex traits. And there's a difference between simple traits and complex traits. So simple traits, the expression of the phenotype, which is a measured level of the performance for a trait in an animal, will be influenced solely by the genetics, right? The genetic makeup. And that genotype will be from a few genes. Now, when you've got complex traits, now the expression is now dependent on the genetics, many genes, and the environment. And Dr. Andy Herring, I think is his last name, hopefully I'm saying it correctly, touched on that yesterday, right? The fact that the environment plays a role in the expression of complex traits. And most of the traits that we're interested in are complex in nature. And this is where the use of EPDs comes in, is on those complex traits. Let's see here. Going back to what we talked about earlier, producers submitting data, contemporary group information, individual records, pedigree records, and progeny records to the breed associations. The breed associations will then send that information to the genetic evaluation companies, and the genetic evaluation companies will, quote unquote, do their magic and extract anything that is non-genetic from that formula and calculate first estimated breeding values, right, EBVs, and EBVs are used in Australia, and then we'll divide that by 2 and use expected progeny differences, EPDs, which is what North Americans are used to seeing. And estimated breeding values are the genetic merit of the animal, where expected progeny differences are the genetic merit of the animal as a parent. So there's a difference between the two. And it's, I don't ask me why we decided early on to use EPDs as opposed to EBVs, but you know, if you're buying a bull, it's not a lawn ornament, you want them to breed, and so that's why, I try to tell breeders sometimes, that's why we use EPDs as opposed to EBVs. We want to know what the progeny is going to have in terms of genetic merit. And that individual animal report is then submitted back to the breeder. So let's spend a little bit of time here on this slide so you know the difference between the two. So the EBV, the estimated breeding value of a progeny, is equal to the EPD of the sire plus the EPD of the dam. The EPD of the sire is really half of the EBV of that sire, right? Go back to this, right? EBV and EPD. And the EPD of the dam, again, is half of the EBV of that dam, right? It makes sense. So it's half of the sire's contribution, half of the dam's contribution. That's going to be the EBV of the calf of that progeny. Andy talked a little bit about this, and that's heritability. We cannot talk about complex traits and not talk about concepts like heritability and even accuracy, which we're going to talk about in a little bit. Again, the phenotypic expression is equal to the genetics, the many genes, not just a few genes, and the environment. And we parse out the genetics component into the additive genetic effect and the non-additive genetic effect. And the non-additive genetic effect is the effect caused by gene combinations, things like dominance effect and gene interaction. And the additive genetic effect, which is what we're interested in, is that kind of effect that is transmitted from generation to generation. Right? It's the kind of that we can estimate and make predictions about when it comes to future generations. What heritability measures, okay, and again, heritability is the additive genetic effect, okay? So it's part of the phenotypic expression that is solely due to the additive genetic effect, and that's heritability. Now, heritability, another way of looking at it is it measures the strength, okay, of the relationship between the phenotypic values, what you could observe, what you can see, and the breeding values for a trait in a population, okay, the genetics. It's not a value to be associated with an individual animal. It's trait specific. So remember that breeding values and phenotypic values, right? The correlation, the strength of that relationship is heritability. And a myth that I still see around is that people think that because a trait has low heritability, it cannot be genetically improved. Nothing can be further from the truth. As long as there is genetic variation, you can improve the trait. The fact that it has low heritability, I mean, it's going to be a little bit harder to, because you now have to collect more observations. But heritability does not tell us, how much a trait can be improved. If there is variation, you can improve it. With low heritability, problems that you have to have more observations to reach a level of accuracy that high heritability traits will achieve with less observations. So unfortunately, some of the economically relevant traits sometimes have low or moderate heritability. So low heritability fertility, for example. Dr. Cliff Lamb yesterday talked about fertility. Very, very important trait, and it should be part of a selection index. And I'm pretty sure that once you start collecting more data on fertility and RFI, so RFI is another very important trait that needs to be part of a selection index. Once you've got enough data, you're probably going to have to go back to a selection. You're going to have to go back to a selection index and add those traits in there, right? So let's look at a visual representation of EPDs, right? Performance information from the individual, performance information from the pedigree, and often we talk about pedigree in the sense as parents and half-sibs, plus performance information from the progeny. All of that behind the scenes will allow you to calculate EPDs. Of course, what I'm not mentioning here is the need for contemporary groups. It's the cornerstone of a genetic evaluation. Make sure you can compare apples to apples, okay? So when a breed association staff asks you, what are your contemporary groups, it's not because they want to be annoying. It's because that is very important to calculate a genetic evaluation run. It's not because it's extra stuff there, an extra column that they just want to have in a database. It's very, very important. Another term that I'm sure you've heard before is called accuracy. Okay, accuracy measures the strength of the relationship between the predicted values, right, the estimated values, right, sometimes use the word predicted or estimated as interchangeably. EPDs, so EPDs are an estimation, they're predicted, and accuracy is that relationship between the predicted values and the true values. Right? And the higher the accuracy, the higher the strength of that relationship between the predicted and the true values. And what you want is a high accuracy. If you can get a high accuracy early enough in the animal's life, great. You can make a decision right there to use that animal as your breeding stock when that animal is old enough or not. Or call it right away. Another visual representation here is the confidence that you have that you're picking the right parents for the next generation, right? You've got more confidence with higher accuracy that the EPDs will lie within a specific range that is definitely smaller than if you had lower accuracy, smaller accuracy, less confidence, right? If you're at the top there, you've got a smaller range of EPDs, a smaller range that the EPDs can change over time, but if you've got a low accuracy, you've got a much larger range that the true EPDs will lie. Okay, that's one of the reasons, and I didn't want to put the formula in here. It's one of the reasons that high accuracy allows you to have a high rate of genetic progress, because with high accuracy, you are now closer to the truth of that EPD than you were before. So you're picking better parents for the next generation, right? You're not guessing anymore. It's more precise, right? And another reason that the rate of genetic progress increases is because you're now picking better parents early on in a life cycle, right? The generation interval lowers, and when you lower the generation interval, increase the rate of genetic progress. So we spent a long time, about 30 years, just doing traditional genetic evaluation. This was way before I was born. And about 10 years ago, when we really cranked up our notion and our knowledge of genomics, we now had a lot of data to work with, and we needed to figure out at this point how to incorporate DNA information into the traditional genetic evaluation. So now a big piece of that visual representation includes genomic, right there on your right. You've got performance, you've got pedigree, you've got progeny, and now you've got to deal with genomic information, a lot of it. Right? And genomic is nothing more than another word for DNA. Right? So when you hear somebody saying, "Oh, I'm using genomic information," all they're saying is that I'm using DNA information. Right? But there's another type of, I guess, genomics, and so those are the genomics-based tests. Right, so you have to be aware that just because somebody says I'm using genomics, it could mean that they're just using parentage testing. I mean, parentage testing is a genomics-based test, or they are actually using the full genomic profiler, right, with those chips, with high number of SNPs, right? You have to make that distinction, particularly if you're out there, selling a bull, and you're talking about genomics with your clients. You have to be able to make those distinctions of the jargon, right? The vocabulary. Are you using a full genomic profile, or are you just using parentage testing, right? And now we have genomic enhanced EPDs. I want to also let you know that genomic enhanced EPDs, it's not, we're not making the EPDs better in the sense they're not more positive, so that's not what genomics does. Again, what genomics does is it increases the accuracy, so it's more reliable, right? It's not making EPDs more positive. That's not what this is about. I like to tell clients too that it all starts with a sample, okay? If you're going to be genomic profiling your animal, you have to have a physical sample of that animal. So you can choose to use a hair sample or a blood sample or a tissue tag. I don't know exactly what BBU accepts. Probably, I don't want to speak for the association, but any of those types of samples, so hair, blood, or tissue tag, would be a source of DNA for the test. And of course, since I'm in Texas, I found this picture. And this is the Nitro Boost that I was talking to you about, right? How fast genomics has allowed us to make genetic progress. Here's a nice little graph, and the dairy industry is really way ahead in terms of genomic use of genomic information. Right there in 2009, you can see a clear line. Right after that, look at that peak right after 2009, and the rate of genetic progress has really skyrocketed. The dashed line that you see at the bottom is if they did not use genomics, that's what their genetic progress would be. Okay, but with genomics, it's increased for both protein and LPI. The Canadian dairy industry uses LPI as opposed to TPI. I don't know if you're well versed in the dairy industry, you're probably used to looking at TPI. I talked about prediction equations already. Now this is the part of the presentation where I go a little faster, I think, because I think I covered the bases. So prediction equations are breed specific. We already know that. Early on we thought we could just have one size fits all and nothing can be further from the truth. And what prediction equations allow you to do is to create a map file, right, that assign the raw data to the correct predictions, right, where we know that an A, a base, a nucleotide, will be equal to, a 1% increase in calving ease direct. Right? So that's all prediction equations are, is a map file that allows you to assign the right effect of a genetic marker to the correct trait, right, and to the correct base, so that you can make predictions later on an animal that does not have any phenotypic data. Overall process, you'd have a calf with low accuracy. You take a physical sample, a tissue sample, you send that to the lab, they'll do the genotyping process, they'll have the data, and they'll send it to the genetic evaluation companies, which then have the specific prediction equations, and they will put it together into the genetic evaluation to have genomic enhanced EPDs. That's the overall process when you're trying to summarize it. Let's talk a little bit about the strategies. I mentioned some of them already, and I'm going to keep repeating myself if I have to, because I know how important that is and how much improvement I have seen and we have seen through data on just using parentage testing. I published this not too long ago on the website. If you have a chance to go read it, please do. And it's called the 3 Golden Rules for Stress-Free Genetic Improvement Program. And stress-free is a bit cheeky because every time you implement technology in a breeding improvement program, it's not really stress-free. But if you follow the rules and the process and the methodical way, it's a lot less stressful. Because at this point, you're getting hit with a bunch of information from all sides. You hear genomics, you hear genomics-based tests, you hear chip numbers and size and names, and you start wondering, what do I do next? So it's the shiny, object syndrome that you're getting hit with at this point, right? What do I do next? But your first step is this. Parentage testing is the first step. You got to make sure that you've got the right pedigree and the right connection of animals in your database before you start even thinking about, a high-density chip. Right? Parentage testing is the first step. And many breed associations have implemented this for years, have been implementing parentage testing via DNA, of course, for years. And it was via the old technology called microsatellites, or STRs. You may have remembered them. And they've switched over to SNP technology, SNP technology. And the sole reason, one of the reasons I should say, is that there are no high-density chips with STRs. There are only high-density chips with SNPs. And the high-density chips are what allow you to incorporate a significant amount of DNA information into the traditional genetic evaluation. So in an effort to condense the work, and many genomics companies incorporated parentage testing markers on those high-profile chips. So you don't have to take 2 samples from an animal anymore. You take 1, right? And everything is done. The second point here is to check all the boxes when selecting a service provider. Remember the picture that I showed and the power, how important laboratories have become over time? Well, you have to make sure you're partnering with the right one, okay? And most breed associations will choose to have 2 service providers as part of on their roster of genotyping service company. And the reason for that is it allows the overflow to be sent to various ones. So make sure that the breed association works closely with the laboratories, the representative, so that you can be aware of any changes and you can have any tweaks that need to be made as you start increasing the number of samples submitted for your genetic evaluation. And third, but also very important, last but not least, is have a communication plan that includes training, right? Many breed associations now outside of the United States are starting to use genomics, or starting to want to use genomics, and they hear how successful some of the breed associations have become, and they call us up, and they say, I want to start using this. And I go and ask them, have you done parentage testing before? I said, not yet. And so we have to go back and say parentage testing is the first step. So when the company found out I hold a Brazilian citizenship, and they shipped me off to Brazil and to try to help out those folks down there in areas of Latin America as well. So many of your customers are in Mexico or in other parts of Latin America. So you have to be aware that what you do in terms of genetic improvement is going to affect them. So anytime you have a chance to help your customers succeed, if you're doing already a genomic profile, a high-density genomic profile that includes the parentage markers that I talked about, you can tell your customers, look, buy a bowl for me that's been genomically profiled, and I'll give you, as a bonus, the genetic markers for parentage, right? Simple parentage markers, because then they are saving about $20 and not having to genotype the animal that you just sold them for simple parentage testing. Okay, so that's another way you combine technical and marketing so that you can sell your products, right? So that you've invested in a product, so now it's time for you to recoup your investment. Okay. I talked a little bit about this. So genomics means DNA, but you also have to be aware what kind of DNA test or DNA procedure you're using. So typically we have three categories, and that's first is parentage testing, it's parentage verification. The second is simple tests, right? So for traits and genetic defects. And the third is genomic profilers, right? Already talked about that. So 50K or GGP, all of those. Here's a very first strategy. Parentage, I'm going to keep repeating myself. You have to perform parentage testing with the right bull battery. You can't just not record your bull battery in breeding groups and then just expect the breed association to do all the work for you in terms of assigning the right bull and the right sire. The problem with just doing a blast search, if you haven't kept records, is that the closely related your bulls are, the more problems you're going to have in trying to distinguish the true parent. Okay, so when you keep breeding groups, the easier it is for the assignment of parentage. Okay, so keep breeding groups and only test the calves of that breeding group with the bulls that you used. Even if you're doing multi-sire breeding, you just keep track of the bulls and the bulls' IDs and say, I'm going to be testing these bulls with, this progeny group rather than all of the progeny groups. What parentage testing will help you with is in setting the two boxes there, right? Pedigree and progeny, right? Typically, we say pedigree to say progeny as well, and typically I do that, excuse me. But in the sense of the way that I have the boxes there, parentage testing will allow you having the right pedigree, which means parents and half-sibs, and the progeny. EPDs, PTAs, if you're in the dairy industry, or EBVs, if you live in Australia, without properly calculated EPDs, PTAs, and EBVs. That is the foundation. Okay, you pour the foundation first before you build the first floor. And remember that a young animal, progeny, will rely on its parents' genetic merit value. So if you have the wrong pedigree, you will have the wrong EPD for that animal that you're trying to sell. This is particularly true, important for seed stock breeders that are trying to sell, yearling bulls. Okay, so parentage testing using DNA technology will help fix that. And I've already alluded to this, having the wrong pedigree can cost breeders thousands of dollars in decreased bull prices and sales. And this is particularly important if you're a seed stock producer and you're selling yearling bulls with limited performance information. And so you're relying on the parents' information. And commercial producers can significantly add value to their herd with parentage testing. So parentage testing is not just for the seed stock producer. Parentage testing is for commercial producers as well, particularly if commercial genomic panels are not yet available for them. And here's some of the other benefits that parentage testing will give you. And that's a really simple way to identify the sires with low progeny numbers. Right? It'll identify sires that cause calving problems. If you're seeing that in your herd, you'll be able to, doing parentage testing, you'll see where the problems are coming from. And third is identifying heifers from the most maternal sires. Okay, so there's some of the benefits of parentage testing along with all the other benefits that I talked about. Here's a very important aspect of information on parentage testing is the type of panel that you use. If you're raising Taurus, you can use a specific panel, and that's typically 100 markers. And these are panels that are approved by ISAG, which is the International Society of Animal Genetics. If you're raising Indicus-influenced cattle, you have to use 200 markers. And again, those are based on recommendations from the International Society of Animal Genetics. So make sure you're ordering the right panel for the breed that you have. Because if you don't, you're going to have problems assigning parentage. Second strategy, I've already talked about this, is only use bulls that have been genomically tested, okay? Because of all of the benefits that we talked about in terms of accuracy, increase and decreasing problems that you're going to have in your herd. Higher accuracy is lower selection risk. Low accuracy is higher selection risk, I should say. This is a typo there. We have GEPDs. I already talked about this, the type of panels, different technology companies. So how do you break away from the pack? Now everybody is using genomics these days here in North America, the United States and Canada. But how do you break away? So how do you differentiate yourself from the rest of the pack? Of course, you have to continue to use genomics. That's not going to, everybody's already doing it. But you're not doing it just because of what everyone is doing. You're doing it because of the benefits that it gives on a breed improvement, for breed improvement programs. What I often tell people, and I alluded to that and I talked about it in the future of the breed associations, is the need for data collection, right? But not just any type of data that will be used to differentiate your breed improvement program from your competitors, and that includes fertility information and RFI. Those two important traits that were talked about here yesterday. I got this from as a screenshot from the Beefmaster Breeders United Facebook page. Tom, we talked about Facebook. I was actually going to quiz you and ask you if you could identify, because Tom said he doesn't have a Facebook, or he doesn't manage Facebook. His son manages the Facebook page. And I thought of quizzing Tom, but I gave it away. It's Facebook. And what this says is that now Beefmaster is now in the middle of collecting feed intake data, and that's very, very important. So continue to do that and encourage breeders to do that as an association. One of the last thoughts here before we close up, I know I'm running a little bit out of time. Now, products will make you money, but systems will make you a fortune. Okay? What is genomics in my view? Genomics is a system. Okay? It's a system that includes phenotypic data collection, the right genotyping panel and service provider, the right genetic evaluation service and method, and the training of end users. And if you skip on any of those, you're going to have a big problem, right? So learn to be part of the system, because with the right system and flow, your breed improvement program will really be differentiated from the rest of the competition. So that is all I have for today. I hope you've enjoyed the talk and you've learned a lot. So if you have any questions that...
I'm certainly not educated, but popular sire syndrome. I've been a dog breeder for many, many years, and we have problems with genetic issues that come up with various strains in our breeding programs. And I know Angus has faced this. I know that I think that you showed those increases in whatever trait was, has had tremendous problems with fertility. They've lost. Are we at risk by following the popular sire syndrome of causing a train wreck? Because there are many very genes that we're not measuring. And so do our set of EPDs, like T-scores obviously are important, dystocia, are these things that we're if we measure them with what we have today, it will prevent that trait run.
Yeah, so the short answer is yes. If you measure those traits that are important for your breeding improvement program, fertility, you can prevent a fertility problem in the future because you can adjust the model. As you see, fertility problems increasing, right, as we saw with the dairy industry years and years ago, and now they've adjusted and they corrected those problems because they saw that the fertility was an issue and it wasn't being corrected. The problem was those, what we call antagonistic markers, okay? And I can't remember who said it, was Andy maybe who said antagonistic markers, ones that have a negative correlation, okay? It will cause negative effects on the traits that you're trying to improve. Okay, so by measuring those traits alongside with everything else, one example that I can give here is RFI. Right? If there is a problem with RFI and fertility, then we have to correct for those so that we're not inadvertently selecting low RFI animals that are very efficient but have a fertility problem. The net effect is zero, or probably worse, actually. So yeah, very, very good question. So the more markers we have and the more phenotypic information we have, the more we can correct for those problems early on. Any other question?
Both of the parents genetically enhanced values, offspring's value to be a little more accurate than it based without.
Oh, you're saying that if you have your parents, the parents have been genomically profiled, does that mean that the progeny does not need to be genomic profiled? That does not mean that. Is that? So accuracy is based on the sources of information. So if you've got, even though you've got parents that have been genomically profiled, it only means that the EPDs of the parents are going to change less than before. But the progeny will still have low accuracy because if you only have the parental average, the progeny will have low accuracy even though the parents have been genomically profiled. Okay, that's a very good question because people typically think it's something that's carried over to next generation. So if you do the parents, you don't have to do the progeny, and that's not true.
In the panel line, Brian's Taurus have 100 and Indicus have 200.
It was just how, good question. I get that, sometimes I get that question too. Just how the markers were kind of identified in terms of variation within that type of cattle. So Taurus-based, we decided that with 100 was enough to distinguish parentage. With Indicus, there's a overlap. So the 200 markers that I have, 100 of those are part of the Taurus. And don't quote me on this, but that's actually, I'd see as a pretty good thing that there's an overlap, because if you're crossing Taurus and Indicus, you can now utilize some of those markers and you don't have to do a completely different panel. So I see that as a very good thing, as an overlap. But with some of the Indicus breeds, and 200 is not, it's the current panel, but it's not set in stone. Unfortunately, I can't stand here and tell you that you don't have to worry about a different parentage panel in the future, because we keep discovering more markers for other Indicus breeds that are less known. So Andy talked about Gir and Hindu from Brazil. The parentage panel works a little bit for them, but it's not perfect. So I would not be surprised that in the near future you see that going to maybe 500 markers as opposed to 200, because of the number of different Indicus influenced cattle that we have. And we haven't even touched on other breeds like Sardo Negro or other breeds in Africa too. So yeah, I think this is just the very beginning, unfortunately. I wish I could tell you that we've got the panel figured out, but it's not true.